So I was talking about this kinetic reading function before my last narration was cut off. So what it means is, it depends on the speed of how this newly coming nucleotide is incorporated into this site. Okay, so in the active site of DNA polymerase, if it is the right nucleotide that forms the correct hydrogen bond with what it's shown on the template strand, in this case A, so yeah, there's going to be a smooth transition. That's nucleotide coming in without any problem, fine transition, and then the DNA polymerase can cut pyrophosphates out and make the phosphodiester bond without any problem. However, if it's not the correct nucleotide that should come in, instead it's something awkward, then the hydrogen bonds are not formed. If there's no hydrogen bonds formed, then this base will be sticking out instead of going in there, right? If that's what's going to happen, and this is what's going to happen. So if base is not forming a correct hydrogen bond, it's sticking out. When it's sticking out, then another part of this DNA polymerase has exonuclease activity right here. Okay, so this exonuclease activity, meaning what? Exonuclease means what? Cutting off the nucleotide from the end of the DNA. That's what it does. So this site has a very strong attraction, maybe 10 times more attraction of the DNA. So it's going to pull that strand over here. Okay, since it's just wiggling out, hanging out there, it's going to pull it out and bring it there, and then it's going to cut it out. So that's what exonuclease would do, cut off the last nucleotide. So that's a kind of proofreading, and if it's incorrect, it's going to cut it up and make it redo, make that tail available again. So the replication polymerization should be redone by the DNA polymerase. That's how the proofreading works. Okay, so he explains right here, mismatched DNA, that's hanging out. So the template primer junction is kind of destabilized because the hydrogen bonds are not formed. If it's hanging out there, then exonuclease activity site will bring it because it has very strong affinity for this hanging out nucleotide, and it's going to cut it. That's how it fixes incorrect nucleotide added on the polymerization site. It's a proofreading function. Okay. And then the replication fork. So when there's a replication fork here, so it's going to go that direction, it's open up continuously, right? There's another replication fork going this direction, you know that. And between these two, this is a leading strand replication. There's no problem because it starts from the 3' end of the primer, it goes continuously. As it opens up, it's going to go continuously. There's no problem. So it's a continuous replication, right? But the problem is right here on the lagging strand, because the 5' end of newly made strand is right here, although leading strand had right here. So polymerase only goes from 5' end to 3' end of the newly made strand, right? So if it's that case, then 5' end of newly made strand is here because the primer is right here, and it's going to go this direction. Okay, if that's the case, then helicase opens up continuously, then this will be left open. It's not copied again, so there should be another polymerase coming in and then do the replication. So it's going to be a discontinuous replication, right? So then what if, the trimer of polymerase, this one, and another one is sitting there together, held together, if that is responsible for replication of the entire thing, how can that trimer complex move along on the template strand together? That's a question, right? The answer for that question is this. That trimer of the DNA polymerase, the hollow enzyme, works like this. There's a leading strand that's being replicated by that monomer there, okay? And then another monomer sits there, okay? And maybe doing replication, single-strand bound protein, but the single-strand bound protein, right? So there's a primer there. It's replicating, so it came from here to here. It's a stretch of Okazaki fragment, if you will. Okay, and then when it's doing it, there's a third polymerase that's sitting there, which was not showing up there, but coming in there. Okay, so when it goes out, there's a third one comes in there, and then it starts. As soon as it opens up, and there's maybe this much is newly opened up, right? Then there's a primer that's just made. And then to the 3' end of the primer, the third one is grabbing the tail, and it's going to continue replication. So between this one and this one, there's going to be a switching out made. Okay, So whenever they switch out, there's a Okazaki fragment length of primer is just being made. So that's always from 5' to 3' end, 5' to 3' end. It's going to go. The switching out between these trimers will take place continuously. So that's how this entire trimer hollow enzyme complex can go continuously through the same direction. Okay, for the Okazaki fragments, 2' units are switching out their jobs continuously. That's how you can explain the lagging strand replication. And then finally, yes, DNA polymerase enzymes have specialized roles in their cell. Yeah, there are multiple DNA polymerases. I think lecture 1 we talked about, the DNA polymerase 1 in E. coli was the first one that's identified. Okay, although that's not the hollow enzyme responsible for the entire DNA replication, that there was another one there. So actually in E. coli there are 5 DNA polymerases that are functioning in the living cells. And we know DNA polymerase number 3 is the main DNA polymerase enzyme, has the trimer complex. And then there's DNA polymerase 1, identified at the beginning. Okay, it was responsible for the repair. Okay, so it specializes in removal of the primer removal. Okay, and then some other repair responsibility activities. Okay, that's what DNA polymerase number 1 does. Okay, and then other 3 DNA polymerases in E. coli are specialized for DNA repair, and they don't have pro-breeding functions. Okay, so they do repair work. So because DNA repair is needed, because DNA could be damaged, and once the damaged DNA are not repaired, then you can create mutations. There are a lot of DNA repair mechanisms exist in living cells. So these enzymes are playing a role there. And eukaryotic cells, and there are a lot more than 5. There are actually 15 DNA polymerase enzymes that are identified in many eukaryotic cells. They are named following Greek letters. Is it delta, epsilon, alpha, and so on. There are 15 of them. Okay, if you look at them, yeah, E. coli, there are pol 1, 2, 3, maybe 4, 5, total 5 enzymes. So polymerase 1, primary removal and DNA repair. That's what they do. Pol 2, DNA repair. Pol 3 is the main replication enzyme. Pol 4, DNA repair. Translesion synthesis, TLS. We'll talk about that later. And DNA polymerase number 5 is a special one. It's involved in the SOS response. We'll learn about what SOS response is later on. And the eukaryotic polymerase, there are 15 of them, from alpha through whatever the last one is in the Greek letter. I cannot read all these, but yeah, there are so many of them. Okay, so each one has maybe different activities and responsibilities. Okay, so it seems like polymerase epsilon is the leading strand in a synthesis enzyme, and is that alpha, beta, gamma, and that's probably delta. Delta is responsible for lagging strand in a synthesis. Okay, so yes, so the rest of them have different functions. Okay, so if you look at them, yes, alpha right there. Okay, and RNA primers are made. Okay. Alpha is involved in grabbing the tail, and then delta and epsilon comes in, and they're switching out. Polymerase switching is happening in eukaryotic cells. That's what it shows. Okay, alpha comes in first, and then the switching between alpha and delta and epsilon. So that's what it describes. Okay, and among other things, also there's a special protein complex called sliding clamps. Okay, that plays an important role during the DNA replication. Especially they provide increased DNA polymerase access processivity. What does that mean? So we're talking about these kind of donut-shaped protein complex that are kind of attached with the DNA polymerase complex. Okay, so we call them sliding DNA clamp proteins. They're made up with multiple identical proteins, make a donut-shaped structure, and they are sliding on the DNA through the gap created by kind of water molecules. Okay, so the clamp protein slide along DNA while tightly bound to the DNA polymerase like this. Okay, the association with the sliding clamp protein prevents the polymerase from diffusing away from every 100 nucleotides. So if it's a DNA polymerase alone, it can just diffuse away after 20 to 100 nucleotide. But this sliding clamp is holding on DNA polymerase so it doesn't fall off. Okay, so since it provides kind of stability for DNA polymerase, so it can hang on to the template DNA. So there's more processability of the DNA polymerase during the replication process. Okay, so sliding clamps are opened and placed on DNA. Okay, and then removed from the DNA by a clamp loader. Okay, so the presence of double-strand DNA in its active site. And polymerase changes conformation and loses affinity for sliding clamp proteins. So, understand sliding protein plays an important role for DNA polymerase to stay on the template DNA. And then DNA synthesis at the replication fork. So, I told you the hollow enzyme in E. coli, there are trimers there, right? So 1, 2, 3 trimers. So leading and lagging strands are synthesized simultaneously. Like I told you, replication fork. E. coli polymerase 3 is a hollow enzyme. There are 3 copies of the DNA polymerase existing. Okay, they are held together through delta protein. So that delta protein is holding those trimers together on that sliding clamp loader and sliding clamp and sliding clamp loader. So that's how these trimers are staying together, showing up there. Okay, and the lagging strand synthesis, of course, is done by these 2 subunits, 2 polymerase complexes. Okay, so you can take a look at this animation, if you will. So you can copy and paste this website address on your browser, and you will see this YouTube video, which is a nice animation of what's happening inside of a cell. It shows you a bunch of different structures that are kind of, I guess, representing different things that are shown there. Let's see if you move fast forward. There may be other things like DNA structure we talked about, right? Nucleosome, histone, and all those things. It shows you that as well. It may show you, yeah, that's probably histone binding and forming the nucleosome structure. If you are interested in seeing that formation, you can do that, as well as, let me see if there are other things. Yeah, see the nucleosome twisting around the entire chromosomal sequence, and then replication is somewhere around there, about 2.5 minutes. Yes, see that? The leading strand synthesis and lagging strand synthesis, this continuous one, is showing up there. Yes, that's what you want to see. Okay, the animation between those 2 subunits, the switching out is made. Okay, see that? Smooth transition between those 2 subunits. They are shown up there. That's the discontinuous Okazaki fragment replication, lagging strand replication, if you will. Okay, you can always do a YouTube Google search with your word like DNA application or something, then you will be able to find these maybe helpful resources if you want. Okay, so I'm going to stop that and go back. Okay, so that's that. And then finally, so DNA replication in eukaryotic cells also require 3 polymerase enzymes. There are trimer in eukaryotic cells as well: alpha, probably delta, and epsilon. So alpha is the leading strand. Epsilon is the leading strand, but between alpha and delta, there's a switching out happening. That's that. And then finally, at the end of the lagging strand synthesis, there's a short primal length of template DNA, about this much, is never replicated in the lagging strand synthesis. Okay, so the reason for that is because there's no enzyme that can synthesize that segment. Okay, so always that primal length, whatever that is, about 20 nucleotide long size, okay, is left uncopied on the newly made replicate strand, on the especially lagging strand. Okay, so every time replication takes place, you lose that much primal length of size uncopied. Okay, so you can see that chromosome is shortening every time it replicates. Okay, so I'm going to stop this and add